Okay, so it's a pleasure today to have Lev Tsimring. Lev is Associate Director at the Biocircuits Institute of the University of California in San Diego. He's also co-PI of System Biodynamics Lab, and his past and current interests include chaos and synchronization, granular dynamics, pattern formation, and quantitative biology. Today, he will talk about floral patterns in multi-strate bacterial colonies. Thanks a lot, Lev, for being here, especially considering the very early time you're giving this talk. So
this is the first time I'm giving a talk at 7 o'clock in the morning local time. But
thanks a lot
like
about
I'm traveling
that.
actually to Europe and maybe giving a talk with a jet lag, so that's okay. It kind of reminds me of some of the previous
the
talks.
Travels and the conferences. So good for that. Yeah. So right. To you, Lev, thank you.
Let's hope it comes back eventually. All right, so yeah, thank you so much for inviting me. It's a pleasure to give a talk at the International Symposium and Dynamics Days. And yes, so you mentioned my interest. So you didn't mention my first interest, actually. So my PhD was in fluid dynamics, and so this was so I'm interested in that in Navier-Stokes equations and things like that. And so this was interesting because this work, which is seemingly kind of has nothing to do with that, actually brought back the memories of that. And so you will see that when I will describe one of the models which we used for this. So the kind of the overall message of this talk is that in biology, sometimes physics and fluid dynamics is also important. So it's not just biochemistry, which is we usually associate with like intracellular dynamics and intracellular signaling. So yes, so this is the work which was done in collaboration with several people. I listed them here. So the most of the work, like nitty-gritty work, was done by my grad students who already graduated, Liang Xiong. He's a physicist, physics PhD. Rob Cooper is a biologist, actually, and so he was helping us with the biological experiments, strains, and other things. So Jeff Hasty is my longtime collaborator on various things related to biology and synthetic biology, and then two other people, Walter Jan Rappel and his postdoc Yansheng Cao. They joined when we started thinking about using the phase field modeling for describing the structures which we observed, and they have done a lot of work on phase field descriptions of cell morphology, motility, and stuff like that. So they were very useful to help us in that regard. So okay, so let me okay. Right, so bacterial patterns, it's an old subject. I actually was interested in that since, I don't know, let's see, more than 20 years already. So I was interested in pattern formation generally even before that, but in more traditional physics areas like convection and Faraday ripples and stuff like that. So when I actually not soon after I came to UCSD, which was in 1992, we started working with Herbie Levine. I don't know, some of you may know him. And Eshel Ben Jacob, who was visiting, and my friend and collaborator Igor Aronson, on modeling of the experimental beautiful patterns in bacteria E. coli aggregation on a semi-solid agar when they were starving. So it's a famous experiment in the lab of Howard Burke, which just came out in 1991. And there were some interesting arrangements of clumps, which clumps were kind of easy to explain because their cells are signaling and they're chemotactic towards the signals which they emit when they're starving. But why would they arrange in those beautiful structures? It was not so obvious, and so we did some work on that. So that was 90. So it was published in 95. So it was already what, 26 years ago. There are many other patterns observed in bacterial colony when they're growing. Some of them are shown here, and the work is actively continuing to this day. Many different systems are studied, many different bacterial strains are studied, and different tools are brought to bear. I listed some of them here. I don't want to spend much time on this, but it's what I wanted to say is most of this lab work is done in monoclonal cultures. So it's a very standard for work in the lab. You take a single strain, you put it on a petri dish, let's say, and just in different configurations and in different conditions, and then you see what happens. And so some strange thing happen, and then people write down equations and model it and try to understand. So, but the life is more complicated than that. So in real life, bacteria exist in communities. We all know about biome now, microbiome now, where thousands of strains coexist somehow, don't kill each other, even though sometimes they try. So the question is, what happens when you have multicultural, multi-strain bacterial colonies? What kind of dynamics you can expect there? And you can expect very complex and very rich dynamics, because there are lots of things that's going on between different strains. They, in many cases, they don't like each other. In other cases, they actually like each other or depend on each other. So some of these typical mechanisms of interaction are listed here. This is the work of our former grad student, Tim Lu, where he just basically listed all possible variants when cells can actually promote each other. So it's commensalism or neutralism, but they don't care. When one is suppressing another, it's called amensalism and mutualism, parasitism and competition. And there are different specific, so this is general idea. So one strain actually slows down the growth of another strain or promotes the growth of another strain. So that's the general picture. But in fact, the mechanism for this can be actually, there are many mechanisms. So some of them are listed here. It can be competition, it can be just mutual competition for food, and they can be some kind of queuing can be involved. Some they can compete for specific metabolite or amino acid, which is in short supply in the environment. They can be cross-feeding, so once they can produce some sort of metabolites which are missing and are needed for another one, they can actually affect chemataxis and this way push other cells out or bring them in. They can excrete toxins. There is this very interesting mechanism which we studied a little bit called contact killing. So this is actually in the bacteria, which I'm going to talk about. This mechanism actually is at work. I mean, it's at work, but it doesn't affect the parents. I will tell you about this. So they can actually kill each other on the contact. They can inject certain, so one bacteria can inject certain, it's like a syringe in other bacteria and inject certain toxin and kill it on the spot. So that's an interesting way of dealing with neighbors. And there can be mechanical interactions. So I actually, this slide is from another talk where I was trying to fit it two works in the same talk. And so I was talking about both of these, contact killing and mechanical interaction. I decided in this talk basically skip the first part because it's just too much. And then just talk about how mechanical interaction can actually lead to interesting pattern formation. So just elaborate the point that there are some multiple mechanisms by which the patterns exist and some interesting dynamics in multi-strain colonies can be observed. I just listed here two examples. One is from our own work on the left. This is when you can actually have three strains. This is the same bacteria, it's just synthetically different. So it only differs by a little bit by different plasmids which you put into the cell. So in this particular case, you have three strains and we call it rock, paper, and scissors. And so the idea is that you can, so again, it is made up that the strain, each strain can kill or suppress the growth of only one other strain, and it does not affect the other strain, the third one, and it doesn't affect itself with this toxin. So it's based on using this three complementary toxin-antitoxin pairs. And if you can see here, some, for example, strain paper, it has a toxin 3 and it has its own antitoxin 3 and has antitoxin 1. And so strain S also has antitoxin 3, so it is not affected by this toxin 3, which is injected by strain P, but it is only strain R is susceptible. So when strain P appears, it started to suppress strain R and so forth. So there's a circle, and this is called rock, paper, scissors system. And you can see interesting sequential dynamics when one strain grows and then gets supplanted by another strain and then by the third strain. So you can have these oscillations in the population dynamics, which we are engineered specifically when you have three different strains. So that's one example. And the second example was, it's another recent paper in Nature Physics by a group mostly from Hong Kong, experimentally at least. So here the idea is that one strain, again, this is two engineered strains, so they're pretty similar except for again, the synthetic component and the synthetic circuits in two different strains are done in such a way that one strain actually produces a quorum sensing signal, which is diffused throughout the environment, and then so this is, for example, this strain, it produces activator A, it produces this type of quorum sensing molecule. It's a small molecule which can diffuse outside of the cell and go back into another cell, and there it activates the production of this protein KZ, and this KZ is the part of the motility machinery which affects the flagella in this E. coli cell. So it actually increases the motility of the other cell. So what it does basically is that if you have a high concentration of cells of type A, cells of type B become highly motile and they basically diffuse out. So there's a high diffusion, and so where there are few cells of type A, then strain B is more or less not motile or much less motile, and there they get stuck. And so now you can see that they actually, by using the schemataxis mechanism, they can push each other out of where they live, and so they start to occupy different positions in space, spatially, because of the chematactic interaction. And so that's exactly what happened. And so if you start the colony from a small spot in the middle, they both grow, but they grow in these rings, and the rings are out of phase. And that's what has been observed, and it's kind of an interesting mechanism. Again, just to elaborate the same point, these are almost the same strains. They only differ in one specific biochemical mechanism, and that led to some patterns. So we wanted to do something different, and well, we actually, the story, I don't know if I need to spend time on this. The story was kind of interesting. So we were looking at these two strains, which are listed down below, Acinetobacter bailey and E. coli, for a different reason. So we wanted to study contact killing, which I mentioned a couple of slides ago. So what this, I actually, in the next slide, which I'm going to spend time on it, explaining what they are. So, but kind of almost by the accident, this student of mine, Liang, he actually looked at a slightly different configuration of the experiment. So he put a mixture of these two cells in a soft, in a petri dish with a soft agar in the center, and let it grow overnight, or like for 24 hours. And what he saw was quite spectacular. Actually, it was completely unexpected. And I will show you the movie. So the movie is with music, so we did it as a promotion for the paper later. So please excuse the over dramatic music in this video, but it's but I think the music, actually I put it to music, so but I think it fits well to what you are going to see. So hold on just one second. So the numbers in the up is the hours. Okay. The credits. So you see, it's 24 hours. Okay. So it's coming. Anyway, so let's see. So this is the pattern basically, which we observed, and it was very robust, very reproducible. So almost, I mean, we started tweaking it, and I will show you more. But it was, I haven't seen anything like this before. It was, if you look at it, it's really strange how it is being generated. It's not ring pattern, it's not sectors like people used to see. This is something else. And so we were really puzzled by that, and we had no idea what is going on. And so we started looking into that. It was really was so beautiful and so interesting, we couldn't pass it up. So just a few words about what these bacteria are. And unlike the examples which I showed before, these are really different bacteria. So I mean, E. coli is very standard. I mean, everybody, at least in my current community, knows what how E. coli looks like. So it's basically a rod-shaped bacteria, which is about 2. I mean, it's from 2 to 5 microns, so it grows and doubles roughly in time, and then in length, and then it divides almost symmetrically, about 1 micron in diameter. And E. coli, like many other gram-negative aerobic bacteria, they have flagella. So that's how they move. That's how they move in liquid. And since they live in intestines, that's basically more or less a liquid environment in most of the intestines, and so that's where they move around. And so they have this very long flagella, which they rotate in the back. And when they rotate it in one direction, it actually creates a propelled motion. Because they have this helical form, if they rotate it in another direction, they go apart and they start to tumble. So it's a run and tumble type of motion. So they can move pretty fast. And but on the surfaces, when they attach to the surface, when they form biofilms, let's say, or just in general, if you put them in a petri dish, they actually lose their flagella and they stop being motile. That's been known. So they really have a hard time walking on a surface when there is no liquid. So that's E. coli. This Acinetobacter bailey, that's a totally different species, totally different genome of bacteria. And so they actually also look like rods, very similar in size and shape, but not always. I mean, it depends on the stage of their life. So they actually change morphology. They call pleomorphic. So they change morphology when they stop growing. So they start to grow to become like bumps. But in the growth phase, which we are mostly concerned with, they actually rod shape, but they have very different type of motility. So they actually don't move very well. So they live in soil and sometimes in water too, but mostly in soil and very often on surfaces in hospitals. So that's why they become very important recently. But not exactly this strain, not Bailey, but another Acinetobacter, but very similar. So how they move on surfaces, they have pili, though they have these little legs, which are shown here, here on all sides, and they move them in synchrony, and so that's how they move. So they can actually walk, basically, and that's what they do efficiently. They don't swim, but they walk. And so on agar plates, which we use, they actually diffuse quite well. So they are still motile. And they also have this interesting feature. So they have this what they call T6 secretion system. So that's exactly what I mentioned before. So they actually kill many neighboring cells on contact. So that's how they compete. And so we were actually originally was setting up experiments to study pattern formation, which is related to this killing mechanism. And that was an interesting study, but this is different. So this is some other examples of this what we call floral or flower-like patterns. So here, these two different strains, they have synthetically, they were labeled by two different fluorescent proteins. Red is Acinetobacter bailey, and green is E. coli. And you can see some amazingly diverse and beautiful floral type patterns. So again, that was what spiked our interest, and we decided to look at it further, try to understand what causes this. So first of all, do we really need two strains to get these patterns? And the answer is yes. So this is the exact same condition. So this is the pattern which we see in the mixture. And this is a temporal evolution of that pattern. So it starts from a small spot in the center, and in 24 hours it grows into a macroscopic. So the size, you can see it, it's 1 centimeter. So it's about 10 centimeter in size structure. So, but if you take just pure E. coli or pure Acinetobacter bailey and put it in a petri dish, what you see is nothing, is quite boring. So E. coli, as I said, is non-motile on the surface. So it just stays basically where you put a drop. So the drop gets bigger, so it becomes like more dense, but it doesn't expand, or almost doesn't expand. And Acinetobacter bailey, it does expand quite well, pretty fast actually, but in a very diffuse round-like shape, which is very common, and most of bacteria do that if you just let them grow from the center. So if they are motile, so nothing particularly interesting happens. And so it clearly, this thing happens when you have both of them together. And you can also actually look at how fast they spread. And the first thing which we noticed is that it's interesting that the pure Acinetobacter is going pretty fast, 3 millimeters per hour. That's the rate of the expansion. And as I said, E. coli doesn't move at all. And when you have a mixture, at least the mixture which was used in this particular experiment, it moves, but it moves about half the speed, slower. So basically, what happens already, that gave us some clue. What happens is that so the E. coli resist motion, so the mixture moves slower, and so somehow I mean, first of all, E. coli also moves. We can see that it's green here. Green color is E. coli. So E. coli propagates with Acinetobacter, but Acinetobacter plays like the role of the driver, and E. coli is basically piggybacking or hitchhiking on top of the on Acinetobacter, and it provides resistance. So they are actually sticking to the surface. So they have to be moved. There is a friction, and so they don't really like to be moved, or maybe they do like to be moved. We don't know, but at least there is some. You have to spend some force to move them around, and so that reduces the speed. And so this hitchhiking, in principle, it's the mechanism which has been known, not in the context of pattern formation, but generally it's known that bacteria sometimes cling to each other, and non-motile bacteria are actually attaching themselves or clinging to motile and can move around using that mechanism. So in principle, it's known. It was known it was not known in this context of forming interesting patterns. So. So just to show you that the patterns are very robust, we tried very different conditions. We tried different relative concentrations of Acinetobacter and E. coli. Well, there is a wide range of about 2 orders of magnitude of the relative concentrations when you do observe patterns. If the concentration of Acinetobacter becomes too high, then you don't observe patterns. So you basically, especially in this case, so there are these magical letters T6, SS plus. That means when the Acinetobacter has this mechanism of killing on contact intact. So it's a wild type bacterium. And so what happens when you mix them together, it actually tries to kill E. coli. I mean, if there is too much E. coli, it cannot kill all of them. There is a finite capacity in this mechanism, so it can only kill so much. So you can still have E. coli and they propagate. But if there is lots of Acinetobacter, they basically kills all E. coli on the spot. And initially, and then it just propagates like there was none of them, and so there is no pattern. But you can also make synthetically, you can eliminate this mechanism, so you can delete a gene which is involved in this T6S contact killing mechanism, and then there is no killing, and so but you still see patterns. You can see actually better patterns. You can see it more in a wider range of conditions, and that gave us a clue that contact killing has nothing to do with this mechanism. So you can have it or not have it. You still have patterns, and even more of them when you don't have killing. So it's a different mechanism. And well, we did some measurements. I don't want to maybe spend time on this. So how the radius of the pattern after a certain time depends on the ratio of concentration, and it's so generally the more Acinetobacter you have, the bigger the radius is, so the faster it moves, which you would expect. But it's interesting when you have the killing system intact. The radius actually doesn't change all that much, and we believe that that's because there is certain auto-regulation going on. So when you change the ratio, Acinetobacter kills the extra, so it kills some amount of E. coli, and so it basically auto-regulates to some concentration, so to a narrow range of concentration effectively. So this is initial concentration ratio, but the real concentration ratio is determined by the quick killing of the extra E. coli in the beginning. So that's why it's more flat and it's actually higher because you have less of E. coli because of the killing and other things. So you can count the number of branches and also count those things, but it's not that important. What's interesting, what's important, and it's kind of obvious, but we still did it. So we can see that the mechanism is actually related to motility of Acinetobacter, because if you basically pull all the legs out of it, so if you knock out the mechanism forming pili, then there is no patterns and there is no expansion, which you would expect, right? It's, so nothing interesting happens either for one for mixture. So mixture works just as well as Acinetobacter alone. And so this is when you very initial. Well, it's just more of the same. Let me skip that. So what's going on? We had no idea. So we decided to look more closely. So we just used a more stronger microscope and just look at the pattern in details. And what we noticed is that there are this. So the structures actually have this very thin strands of E. coli surrounded by Acinetobacter, and they are merging. So the direction of expansion is to the right. So that's so this is behind the front, and I will show you the front in a second. But you see that how these thin lines of E. coli, and there is almost no E. coli in between of them. There is some, but not much. But they are concentrated in these very thin filaments, and they're merging behind the front, and they keep merging. They keep being generated and merged, generated and merged. So there is this constant mechanism of producing the strands and merging them, and that's what creates the floral structures. So if you look at it in motion, so this is the movie, and you can see here. So this is the front. So the colony is behind it, and the front moves to the right. So I will start the movie, and I will try to stop it. Yeah. So you see, this is the front, and that's how it looks very locally. So that's a very local thing. So you can actually see here. So this was the front in the beginning of this particular recording, and you see it already has these things which are left behind, and there's an overall structure which is also kind of forming this dimple on the front, and it gets deeper and deeper and deeper, and it actually forms, and so this front actually folds on itself, and that's where these things are merging, and that's how you get the merging of the structures. And so these things which are behind, they are also formed as the front was folding before. Just so this is a recurring process of the front modulating and merging onto itself. So just looking at this for quite a long time. So this, you see that's what happens. Boom, and this is, so you have this stem, and there are fronts which are, there are these, again, filaments of E. coli which are colliding behind, and the front is already here somewhere, so it's out of view almost. And this process just keeps going, and so eventually cells may be growing behind, so that's how the flower gets filled in. But it seems that the process is started at the interface, at the front of the colony, and that's what gave us a clue of what the mechanism could be. So we just looked at this, and then we realized that presumably.
Excuse me, just if I may, small very clarification. If you know the size, you say it's very local. So what the size about this?
Well, this size is maybe a few millimeters. Yeah. So you could see those. Remember, you could see these stems in the picture, which was 10 centimeters big. So this is not very far away. But you have kind of you have these structures on different scales. So you can have very big ones, and you have small ones which are going into that. So it's really like a leaf. So it's almost like fractal. I don't know if it's I'm not sure if this is exactly fractal or not, but it's kind of like that. So you have the front which already has dimples, but then this front actually has a wider undulations which are also with this things coming out of it. It also falls into itself, and that's how you have these bigger structures and bigger structures, and so on. And the front is propagating, and new cells are cells are also growing. Imagine, so both E. coli and Acinetobacter keep growing, and so that's why this pattern is self-regenerating.
Thank you.
So yeah, so we just looked at it and we quantified it a little bit, and so what we kind of intuitively felt, it also was confirmed by our measurements. So we basically looked at the front, and we quantified the brightness of green on the front, which gives us the measure of how much E. coli you have on the interface. And we also measured the speed of the front and the curvature of the front locally in the same positions. And so what we noticed, I mean, curvature is not particularly conclusive, but what we definitely noticed, there is an inverse correlation between the brightness on the front and the speed of the front. So the more E. coli you have on the front, the slower it moves. Okay, and so and that's what we decided. That's probably the mechanism for this whole process. And that was indeed the case, because I will show you the models where it actually is. That's what's happening. So we're actually at the model. So now let's go to the models. And so we have two types of models for this. So one is very abstract, but tractable and simple and very kind of intuitive. And so I will start with that. So this is basically just an interface model. So imagine that you have an interface, and this interface is growing. And it's growing because there is a force which is normal to the interface, which pushes it outwards. Okay, and so we can write down the equation. So there's a force and there's a friction. So the interface motion is frictional. So friction is actually not constant in this case, but let's forget about this for a second. So the general equation of motion is mu, this is the friction force, so it's overdamped motion, dx dt, x is the vector which characterizes the interface, and sigma is, well, sigma is later. So yeah, so now it's just x. So dx dt is n, which is the normal to the interface, times f, which depends on the position, on the current position, generally speaking, right? So this is the general equation of motion, and you can parameterize it by introducing the coordinate along the interface, which goes from 0 to 1. So along this, it's a closed curve. So now the arc length does not have to coincide with sigma because the interface is actually growing. So arc length is changing. So arc length goes from 0 to L, which is the length of the interface, which is a function of time, right? And so now you can, instead of working in these Cartesian coordinates, you can go to the natural coordinates of the curvature and the tangential vector, or the metric, which is so tangential. So the curvature is the second derivative of the position on the interface with respect to the arc length, so that's the definition. And you can introduce tangential vector tau, which is basically the derivative of tau with respect to sigma, and the metric you can introduce as a dot product of tau. It's a norm of the tangential vector. So if tangential vector becomes longer, so if the interface stretches, your metric gets bigger, right? So the metric characterizes how stretched the interface is locally, all right? And so there is a connection between arc length and the coordinate along the interface. So it's basically the integral of the square root of the metric over sigma. So that now, once you have this sigma, once you have kappa and g, then you can write down the equation, and s, the arc length, you can write down the equation for the interface in local coordinates, which are not related to the position position of this whole thing in space. And it actually was already used in the paper by Havre and David Kessler and a couple of other people very long time ago, when they were describing solidification patterns. So a completely different problem, and the mechanism, and they also had some interesting instabilities there which form dendrites, but it was a very different mechanism, not related to this at all. So there, the force actually was doing some strange things. So it was, instead of being force, usually normal force, when you have a curved interface, it actually, because of the surface tension, it actually gets smaller. So the surface tension tries to minimize the curvature. That's typical. What they did in that paper, they say, what if the interface, if the force actually grows with the curvature, so if it becomes more curved, it pushes stronger, then obviously you have an instability, so the dimple would go forward, and that's how you get dendrites. So that was then that paper. But what we got from that is that the general equation for the curvature and the metric is exactly the same as in their case, is in their paper, so you can actually use this. But in our case, the mechanism of pattern formation is not that. So so what's behind this interface? I mean, it's not explicitly in the model, but implicitly is this growing colony of Acinetobacter. Let's forget about E. coli, which is inside. Let's assume that all E. coli is concentrated on the interface. So that's the E. coli which matters. So the E. coli which is behind doesn't matter. That's the assumption of the model. So, and then now what happens is that if the concentration of E. coli locally on the interface gets larger, then the friction of the which parameter friction mu gets bigger. So that's why if you have more concentrated E. coli somewhere locally here, then it will propagate slower because of the friction. So what happens to the concentration of E. coli? Why would it ever change on the interface? Well, let's assume that it's actually it's a conserved quantity. So we have certain amount of E. coli, certain concentration C on the interface. And then it only changes by stretching of the interface and contracting of the interface. So where the interface stretches, the local concentration gets smaller, and when it contracts, it gets larger. So on the next slide, so the driving force is very trivial in this case, so nothing peculiar there. So it's just a constant force acting from the back, and there is a curvature correction term. So it's just normal surface tension. So if there is a curved interface, it tries to push it back. So it's good to have it to prevent forming crazy structures, because otherwise it can create cusps and so forth. But the friction, but as I said, the friction is actually dependent on the local concentration of the E. coli, which you call C, and it's a linear. We assume that it's a simple linear function, so there is a constant term, and it increases with C. So by what I described before, if you assume that the E. coli is not produced and is not lost, it's just constant on the interface, but only can be stretched or contracted, then it's easy to see that the local concentration is just a constant, initial constant. So initially metric was 1 everywhere. So that was by construction. So, but then it starts to go down, and it goes down with metric. So once we assume that, we have a closed, so we can plug it into the friction, and now we have a closed equation for curvature and the metric. So C drops out, and we have this equation. So we can analyze them. You can linearize it with respect to, like, if you have a very big radius of the colony, you can forget about the overall curvature. So you assume that there is a plane front, and you can create some periodic undulations on the front and see if they are stable or unstable. So you can linearize it with respect to that constant propagation speed of the interface. So, and then you can see that it's actually unstable, like you would expect, because it's kind of clear if you have. So what happens is that if you have a small undulation on the front, the force which pushes it forward is constant. It doesn't. It depends weakly on curvature. So it's slightly bigger. It's slightly less. But so let's suppose gamma is very small, or even 0 for now. So then F is constant, but the friction force here is smaller, because once you created this undulation, you stretched the interface there. You stretched it there, and you contracted it here. So here the friction force gets larger. Here the friction force gets smaller, and so now you have imbalance on forces. So this thing actually starts to move faster, and so this undulation on the front is actually moving even faster. So you have it grows, it becomes bigger, it stretches even more. The friction force gets even less, and so that it runs away. So the front actually in this place accelerates, and in this place it actually gets slower and slower and slower, because E. coli gets more and more concentrated in these places. So this is the place where the cusps presumably would occur, those highly concentrated positions on the interface which would create this, which would initialize this, these lines which we see behind. So for now, we don't have any lines behind. We just have this mechanism of the front instability. And so that's basically what's going on. So the mechanism is very similar to how, the balloons, which you blow the balloon up, then eventually it blows up, and it blows up in a big spot, so to speak. So where the balloon, when the rubber gets very thin, it actually creates a bubble, and the bubble eventually bursts, because that's where the force actually, it's not exactly the same mechanism, but sort of related. So this is what's happening. And I don't. Do you have any questions, guys, about this before I move on?
Yeah. Can I ask you some questions, Rob? So you explained that. So the reason that it doesn't branch is it is really because of the surface tension, right?
It does not. Well. It just doesn't blow up. That's what I would say. So surface tension kind of keeps it sane. So because if you have stronger and stronger undulations, because in this case, so because of this instability is for all wave numbers. So this K here is a wave number of the undulation. So the bigger the wave number, the faster the growth rate is, and so this usually leads to forming singularities maybe in a finite time. So to prevent that, we introduce surface tension, because that's what usually keeps it localized.
But in the case where E. coli is actually getting destroyed, so there's less and less of them, like in the killer case, wouldn't the surface tension get less and less when it start to branch out and form a different pattern?
Well, there is no less and less. That's the thing. Is yes. So killing, for example, tries to destroy E. coli, but E. coli also is growing. So it's not stationary.
So it has to do with the relative growth rates of the two, right?
It does, yes. So if there is, right, and E. coli actually grows faster. So in that sense,
I see.
I don't know.
Even in spite of the killing, it grows faster.
Well, no, yeah, separately it goes faster. So in spite of the killing, that's the question. So the thing is that what happens is that they are, in this case, they're kind of semi-separated. So the cells which are inside of the, surrounded by Acinetobacter, they get killed slowly, not fast. So I don't know if that's enough. I mean, they still remain there. But the thing is that, so the question is why cells are actually accumulating on the front. So why there are so many E. coli on the front? And that's presumably because that's where they can thrive. There is no Acinetobacter there. They're kind of pushing from the back. But E. coli, which are in the front, where they are growing faster, and so they are regenerating there more efficiently than in the back. So that's not. I mean, it's a different level of description, which I'm going to show you later. But it's interesting that E. coli, we don't create a front of E. coli which is surrounding the Acinetobacter. It actually creates by itself, because they are the ones which are escaping, so which are actually happen to be in front of the Acinetobacter. They actually have a better time of growing. They have more space, even without killing. They have better time of growing. They are actually, it reminds me when it's like a snowplow. So they are actually, if there is E. coli and it's growing, so you are pushing them forward. So that's what creates this layer, and in this layer they're growing fine. So they are regenerating. You're constantly losing E. coli in the back. So they're actually escaping from the front. So some of them become stationary, so they don't propagate. But you are generating more, and you push, and the front pushes them and concentrates the front. So
and what about the reason for the merging of the different fronts? Is it because they try to stay round? I mean,
the reason for merging is
right,
actually,
instead of growing out a long arm.
Let me show you the next slide, and maybe you will see.
Okay, thanks.
So yeah, so the reason for merging is actually entirely within this model. So you don't really need anything else. So what we did is we actually, instead of linearizing this model and analyzing this, which is a partial differential equation type model, what we did is we actually simulated a discrete element mockup of exactly the same system, where there are no other mechanisms, just what I described here. So basically, we considered a chain of dots, and each dot is basically like it's like a clump of E. coli. So we have a fixed size, and we fixed the friction. So and they're connected by elastic by springs. So it's a closed curve, and so each ball here on this curve is there are two forces which act on that. So one is the constant normal force to this line, which is F0, and there is another force which is a combination of couple of different things. There is a resistant force which depends on C. So what C is the local concentration of balls. So it's basically roughly speaking, it's inversely proportional to the distance of this ball to the next ball, to two or like a group of its nearest neighbors. So it's a local density of balls on the interface. So that's one component of the resistant force, and another is basically the spring force. So if there is an angle between two neighboring springs, it also contributes it's basically surface tension now. So if there is an angle, the force increases, and also there's a repulsion between balls and neighbors or any other ball. So there's this because otherwise this interface, if you let it grow, then they will start to intersect each other. So we don't want self-intersection. So that's it. So that's basically the model, and we just let it. We integrated it and we let it grow, and you will see what happens here. So you see. So let's forget about the right one. It's not that important. But let's look at the left one. So initially it was a circle with very tiny perturbations of the positions of the balls on the circle, and these perturbations started to grow very rapidly. And see already here you can see it before, but they formed undulations, and when undulations actually grew bigger and bigger and bigger, so they start to force onto each other, and that's when the self-avoidance kicked in. So they don't really overlap like they would if there was no self-avoidance, but they actually got stuck, and they created this like a bilayer. So, and these bilayers, they form this, and they are stationary, so they're actually pushing to each other, and they don't move anywhere. So these fronts actually, see, they are just, and they continue to collide. So this was one basically front, and then this is another front, and then since these parts keep growing, they actually let them to collide and form a single front. See, boom, they form a single front, and So these balls still propagate because there is a constant force F0. The resistance force here is actually becoming smaller and smaller and smaller because the distance between the balls, now we can see them, because the overall length of the curve gets bigger and bigger and bigger, but the number of dots is constant. It's not growing in this case. And so the friction force here becomes very small. And so the pattern actually is only transient. So it actually runs. So this whole thing is running away. So it just keeps growing. And there are no more patterns appearing because now the friction is irrelevant, basically. So the friction force gets smaller here. So new fronts don't form. Because in this model, we don't add regeneration of E. coli. So if we would introduce a new formation of new balls on the interface, that would create self-regenerating patterns. But we didn't do that. So because it's only a conceptual model. So we just wanted to illustrate the mechanism. So the mechanism is there. It just transient in this case. If you look at basically the mark, the position, the concentration of the balls on the interface at any given time, starting from the very beginning. So blue means high concentration and red means low concentration of balls. So you see this beautiful pattern, which is not a real pattern. It's just basically like a fossil record of the motion of this interface in this model. It already looks like a flower, not exactly. And so there are no more, it's different from what we see in experiment because it's only transient, as I said, but it's kind of looks pretty, right? So this is, I can play that too. So that's what happens. It's the same simulation as on the left. It's just a different way of showing what's going on. So you can see that the cusps form, which presumably give rise to these stems in the leaves, to these veins in the leaves, and then they emerge, like in many cases you can see here. So the merging of them is the part of the model. So so that was one model, but we also wanted to look at it more carefully and just introduce all the physical mechanisms, which I mean not all, but like more the relevant ones, which we believe are play the role here. And so we decided to do a sort of it's a partial differential equation type model. It's based on the phase field description. So since we have a localized domain where everything is going, so we didn't want to kind of to let the problem is that when you have cells growing, they start to grow everywhere because if there is diffusion, they propagate outwards, and you have this what is known like a Kolmogorov KPP Fisher type dynamics. We didn't want that, so we wanted to limit the structure by a certain closed domain, which actually can move around and can expand. And so that's why we have a phase field model. So the phase is a fictitious function which characterizes the location of the colony. So there is this so there is this term G of phi. I don't want to go into very big details, which is basically creates a bistable potential for phi with fixed points 0 and 1, and 0 is where the outside of the colony and 1 is inside of the colony. And this phase field can be diffusing with a small diffusion coefficient epsilon, and it also can be transported by the flow if there is a flow inside of the domain. So then where the flow comes from, and that I will get to that. So let's so in addition to this phi, you have the two equations for the density of two bacteria. So I call them red and green. Red is Acinetobacter and green is E. coli. And so they also diffusing. And diffusion is rather weak here because I mean it's bigger for the red bacteria, because they are motile, and very small for green. But in any case, they also grow, and this is the logistical growth of these both grains. They only grow where the inside of the phase, inside of the domain. So that's why phase is inside of here. So, and also the growth is saturated by both of them. So they're coupled in this way as well. So they're coupled because if there is lots of E. coli, Acinetobacter cannot grow, and vice versa. So the total concentration of both bacteria strains reaches 1 in a fixed point, if it's a uniform state. So, but then there is also a drift term. So it's a convective term, which again is transporting both strains by the velocity field. And where the velocity field comes from, that's where the fluid dynamics comes in. And so you just basically write down the Navier-Stokes equation, and so it has this form. So we have a viscosity, which is not that important. We just put it in for to smooth out the dynamics. But the main physics here is the balance between the stress. So the stress is produced by the growing red cells, only by the Acinetobacter. So they actually create the pressure. So they're creating the pressure which pushes things outside. So this is basically the pressure term. And this pressure, the gradient of pressure, is balanced by the friction. So this is the friction with the bottom, and the friction has a constant term C, and it has a green cell dependent term rho g. So, and it's a Hill function. So basically, it's a growing function of rho g. So the more green cells you have, the higher the local friction is, and so with some coefficient beta. So this is basically, so the crux of the model is here. So you have so if there was no rho g, then you would have just some expansion, and that's important. So it's not just diffusion in this case. So cells are actually pushing each other. So there is a directed motion of the cells. So it's really an overdamped flow rather than reaction diffusion type system, which is, that's what is different. You have to keep it in mind when you look at the expanding bacterial colonies. It's not just diffusion. It's actually, the cells are actually pushing each other to propagate. So the cells which are growing in the back, they also contribute to the motion of the cells in the front. So, but here the important thing is that so we don't specify where the green cells are, but they are growing, and if there are some green cells locally, that creates the friction force for the whole flow. So that's the model, and it's hard to analyze it analytically. We did it numerically mostly, and I will show you what happens. So this is the simulation of the growth with pure E. coli and pure I. bali, and as expected, there is nothing interesting happening there. So E. coli doesn't grow because diffusion constant is very small, and Acinetobacter diffusion is higher, and so it expands, and so you can also calculate the velocities. Now, if you mix them together, it actually starts growing, and you can measure the front, and it's actually slower, which is, again, you would expect, because the friction, bottom friction becomes larger if you have green cells. And so this is the phase field simulations in 2D, and this is initial condition. So you have a little bit of E. coli, and you have some, it depends on, you can vary the local concentrations quite a bit, but that's the particular simulation here. And so this is what happens. So you see, you start forming this. So the concentration of E. coli is actually higher here on the interface. So like I said, E. coli actually spontaneously form this dense ring on the interface. But in this case, if they are not localized on the interface, they can actually live anywhere they choose to. And so what you see is that the front is unstable, it propagates, and it leaves behind those branches, which is, I mean, this picture looks very much like the experimental picture. So this is, that kind of convinced us that there are no other mechanisms which we missed. So apparently, as far as we can tell, so all the reason for forming those beautiful flower patterns is just this physical mechanism of the growth of cells and the friction, the friction which is dependent on the concentration of the non-motile component. So you have motile cells, you have non-motile cells, and non-motile, they are transported by the common flow, and they create friction locally, which depends on their local concentration, and that's enough to produce those patterns. So I'm about to finish. So this is another movie which shows you in more details of how the stability is formed. So you see, it's very similar to what we saw in this interface model, but this is a two-dimensional model. So this is convinced us that that model also made sense. So anyway, so this is my conclusion. So let's see. I think I'm slightly over time, but sorry. But there are some questions. So basically, what I said is that different strains, they can produce very complex patterns just because of the physical mechanisms, not because of the biochemical interaction or some engineered killing or suppressing or exchange of metabolites, just because of the physics. And so here we trace this origin of those flower patterns to the front instability, where the front speed is inversely correlated with the concentration of the non-motile strain, E. coli in this case. And so we made two models. One was an abstract interface model where we show that the interface is unstable, and because of the local friction on the interface. And then we created a bigger two-dimensional distributed phase field model, which showed basically very similar mechanism, but also reproduced the patterns much more closely and in the bulk, not just on the interface, but in the bulk it showed these flower type patterns. And so this paper was published last year in eLife. You can look it up if you want to. And again, thanks to my collaborators, which did a lot of work on this. And